Dear students, now I shall explain you about the evolutionary significance of chromosomal aberrations. Evolutionary significance of chromosomal aberrations. Okay? Today I will deliver a lecture on this context to you. Chromosomal aberration are an important source of variation in a population, partly due to position effect and partly due to the formation of chromosomes with a different number and arrangements of genes. Listen again. Your chromosomal aberrations are an important sources of variation. Okay? In a population, partly due to position effect and partly due to the formation of chromosomes with a different number and arrangements of the genes. Translocation produce a new linkage groups. Translocation produce new linkage groups. Inversions help in your preserving gene sequences in different populations, while duplications produce gene families having several copies of the same gene, providing more changes for mutations. And phenotypic variations, similarly in given figure, shown how human chromosome number 3 has evolved by the translocations and inversions in 6 steps. Okay? I explained what is chromosomal aberration, what are the relationship with your population, and you already know here chromosomal aberrations are an important source of variation, no doubt, in a population. That is partly due to position effect and partly due to the formation of chromosomes with different number of and arrangement of genes. Okay, the translocation produce new linkage groups. Inversion help in preserving the gene sequences in different populations, while duplications produce gene families having the several copies of the same gene, providing more changes for mutations and phenotypic variations. Okay, and similarly in figure, I will show you now that figure show how the human chromosome number 3 has evolved by the translocation and inversion in the 6 steps. First of all, you take the chromosomes to explain the step 1. Step 1. Try to understand. Now I shall explain you initially about the translocation. Translocation. You take the chromosome, this is one chromosome, and identify it easily and can consider that is chromosome number 8. Another chromosome also remaining nearby of it, that is chromosome 11. Chromosome 11, and you mark here. Okay, this is your chromosome S, and this is chromosome T, and you mark to find the fragments. Okay, rightly. Okay. This is considered A. Okay. Then in others we mark here. There is another chromosome which will present nearby of it. This will be T. T also be connecting here along with the C. C also be there. Sorry, S. That's C. This is S. S is like here, and for mean. Okay, this type of structure will be there. Okay, and that is called as your translocation. Generally, they can change that locality. The segment will be changed off, and that is generally accomplished in the step 1. That is called as your translocation. S is there, and you find also be T, and then you mark. It will be also be combined here, and the fragment also will link with your T, likely. Okay, but this one is the A in there, in addition to it. Then you mark there, and this is called as translocation. It will be complete first step. Then step 2 is followed by it, the step 1. Then you find the step 2. Step 2. What happened in the step 2? The fusion. In there you find the fusion. That is step 2. Okay? And you consider this chromosome. Okay, this one. See the picture? The A. Then you find another chromosome, and this called as R. Or you can label it lightly. Okay. Then at the end you find such a manner the product will be produced likely It is R, no doubt, and this is your A. You find middle, this portion, remaining here. Okay, the case is known as your future, and it will be accomplished in the state 2. Step 2. Then you find the step 3. Step 3. What happened in step 3? Consider the step 3 here. Okay, you must find the chromosome. Okay, this is called as your R. This is called as your A, and you mark here, you find one segment, additional segment of there. Okay, and then you find in there, they will be also be divided off by the inversion manner. Okay, position also be changed in there by inversion. What happened in there? No doubt you find there the chromosomes. Likely. Okay. And you mark here the chromosome fragment will be remaining in the left side. Okay. And due to what? Your inversions, automatically the part A is going to be off. A is going to be off. Okay. Mark here, A is going to be off, and this is called as your A. Then you mark your R. Okay, this portion will be called as your R. And this is the case of your inversion, called as inversion. Okay, and what happened? The portion which are generally remain within this chromosome will be converted to this form. Next, come to your step for inversions. Step 4, inversion. If you observe the step for inversion, you take here your T. That's another chromosome segment. Okay, and in that segment you find the two segments of the chromosome. This is the A-T, this one is the S. Okay, and here you find the inversions. You find here inversions. Okay. then ultimately you can get one exchange body, and in there you find likely you see that due to inversion change its position. Then this portion is called as your S. Okay. And this is the your what? Inversion in here. And that is called as your inversion step 4. This is known as your step 4 inversion. You can collect some knowledge about it. Then you find the step 5. Step 5. Okay. You mark here the chromosomes also be differentiated off. Okay. And this is the step 5, the case of the fusion. This is the fusion. Okay. And you will find the fragment, and that is called as A. Okay. Then you find your what? And in there you find the what? This type of arrangements. Okay, and this one is called as A, and this portion is named as the your mark here. Then after it you find there one space, then you find second space. Okay. Look the diagram here. These two are left. Lastly, you find the R is there, and this is called as your step 4 fusion case will be arised in there. And do another chromosomes in here. Okay, this is the chromosome. You mark in here. Okay. This portion is known as S, right here. Then this one is the T. You find there. And this chromosomal body has the A and R. Then when they will be fused with each other, what will happen in there? You find an elaborate chromosome. Okay? And you find the segmentation likely. Okay? The next segment is likely. Then the segment R should be there. Then you find T. Let us know what's the segmentation named or read by letter wise. The T. Okay. T is there. Mark it. If they will be associated or should be linked up by fusion, what happened in there? Okay? And you must mark the A must be present at the here, not here, exactly in here. That is the A. Then you find this is the what? R. Okay. After the R, you find the T. With the T. Then at the end, you find S. Okay? And A, R, T, S. A from this chromosome, R from this chromosome. Okay. Then after 8, after taking these two, what? A and R. A and R. You see the A is the R, and you must find in there, and you find the 2 there. Then you find the T just after it. Then lastly you find the S in there. And those segments you have seen by the union of the fusion of the two different fragments. Okay? And this is the product at the end of the step 5. Then come to the step 6. Step 6. In step 6, you mark here the chromosomes also be changed off the positions. Okay? Let us take it. The product one, which one remain like this, and this is called as your A. Okay? And then you find your R. Okay? Then you find your T. After the R, you find the T there. This is the T. Lastly, you find the S. And this portion is called as your S. And this is the product. The same product will be given here. And it will be converted into the one multiple type, and that is called as also be inversions. That will be arising there. Inversion. Mark here. In that inversions, this is the A. Okay. Then you find the R. Then you find the what? S. Okay. Is there? Then you find your R. A is here. Then you find the R. Then after it you find the T. Then lastly find what? Your S. Okay. And then you find your S. Okay. And some peculiarities will be happening in there. The segmental exchange, it will be converted or inverse, and that is called as your inversion. Okay, this case is arised in here, and that is called evolutionary significance of chromosomal aberrations. Okay, thank you all here.